Hello guys, and in this video we'll be discussing gene expression. Basically how we go from DNA to protein. This is our cell that I'm drawing here. I'm going to exaggerate the nucleus because there's some steps that are going to happen. So here, this is our cytoplasm. I'm going to label C. This is our nucleus. I'm going to label N. And then, so basically here in our nucleus we have our DNA, right? And basically it has these proteins that it wraps around called histones, and it could be condensed or decondensed. One is very condensed, is known as heterochromatin. One is loosely condensed, is known as euchromatin. The one that's transcriptionally active, meaning the one that can be made into protein, is the euchromatic form. And so in the DNA there's genes, right? A gene for something. Let's say this is the segment for the gene for, let's say, a receptor on the cell surface, right? Let's say this is for a potassium or sodium channel, right? Or a receptor for, like, I don't know, whatever hormone, epinephrine, right? So you could have a gene in there for a particular protein, and what needs to happen is you need to have a process known as transcription. I'm going to write TS. Transcription. What would be a good abbreviation? I don't know. But yeah, transcription, I just put T. And then you're going to have, well, I'll put TS because it's transcription. And then I'll put TL for translation, which is the next step. That's going to happen in the cytoplasm. Okay? And so what happens is here you're going to have something called the RNA polymerase, which is going to convert this portion of the DNA, the gene, basically the gene which codes for protein, into mRNA, which is single-stranded. And instead of uracil, instead of thymine, you're going to have uracil and mRNA. So just remember that. And that's going to be RNA polymerase. Basically, before this mRNA is able to leave, so the nucleus has these little pores that I didn't draw. That's where the mRNA is going to leave when it's mature. There's going to be some steps that are going to take place. There's going to be a cap that's added to the 5' end, a 7-methyl guanosine cap on the 5' end, and then a poly-A tail. So a bunch of adenine residues. And this is going to prevent the mRNA from being degraded by nucleases in the cytoplasm. The 5' cap is also going to help initiate translation. Another process that's going to take place, you're going to have removal of introns via alternative splicing, which is, so the mRNA has exons and introns. The introns are called, also known as intervening sequences, are going to be removed, and the exons are going to be kept. So here's what we're going to have, something much smaller than what we had there, right? Because you removed the introns, and then you kept the exons. And the alternative splicing means that you can cut it in different ways to get you different versions of a protein. These are known as isoforms, all right? Okay, so you cap, you add a poly-A tail, and you can alternatively splice. Yeah, so those are the 3 steps: capping, tailing, and alternative splicing. Now the mRNA is ready to leave out through these nuclear pores and reach a ribosome. Let me draw a ribosome right here. That's the large subunit. This is the small subunit. Now what's going to happen is the ribosome is going to go through here. This ribosome is going to read it, and these little things called tRNAs, they look like this. All right, they're going to carry amino acids to the ribosome to read this message of mRNA, and then basically make a protein. And the proteins will be made up of amino acids, obviously, and a chain of them. They're going to combine, blah, 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 and then they're going to fold into the structural protein. Let's say in this case, what we talked about earlier, they could fold into something like a receptor for epinephrine, or let's say a sodium or potassium channel in this case, right? Thank you for listening. If you have any questions, please send them in, and don't forget to please subscribe.